One of my favourite films growing up was Back to the Future, and in that there's Dr. Emmett L. Brown. I think he's got a kind of canonical example of a scientist. And there's a line in that when someone asks him, like, he says, what do you do for a living? He says, I'm a scientist. And then he says, what kind of science? He says, well, actually, I'm a student of all science, which at the time doesn't really make, was just a throwaway line in a film. But that's kind of one of the fun bits of doing science, is that our lab is incredibly collaborative. We have biologists, we have chemists, we have physicists, we have engineers all working together to try and solve problems. And for me, that's a really exciting part of the research we do here at Cambridge, is to be able to work in a collaborative team to be able to solve something together that we just couldn't do on our own. We're an academic research lab, so first and foremost, we're trying to understand the way the world works. And we're trying to ask basic questions about new molecules, about new physics and new biology. Chemistry is a kind of central science to a lot of those questions. We're physical chemists, and what we actually do in the lab is we use lasers, and we shine lasers on small chemical molecules, and we do that to illuminate the insides of cells. So we're using lasers to try and understand how biology works, and that's really important. It's got lots of fundamental applications associated with it, trying to understand how to build better machines to be able to do that, but also has really important applications, some of them being disease related, for instance, directly impacting on human health. The lab's split into kind of two halves in general. Half of them are kind of building these instruments I was talking about, and half of them is applying them to biological problems. And so some of the projects we have in the lab are developing optical instruments, so developing fluorophores, developing new instrumentation, and that's predominantly through the use of single molecule fluorescence and super resolution microscopy. And then the other half of the lab is applying those developments to important questions. And the types of questions we're looking at the moment are kind of threefold. We're looking at the molecular origin of adaptive immunity, and that's with collaborators at the University of Oxford. And so that's how your body deals with new infections. So we're trying to understand how your immune cells physically decide whether you're infected or not. And that's not understood, and that requires advanced optical imaging methods to be able to see it. The second area that we're currently working in is in the area of neurodegeneration, so diseases like Alzheimer's and Parkinson's disease. And we're trying to develop new chemical probes that can tell us something about how these proteins misfold and aggregate during the disease. And we want to try and develop the tools to be able to understand those diseases better. And then finally, we're also working on what's called aberrant signaling in cancer cells. So we look at a receptor on the surface of your cells called a KRAS, and this molecule diffuses and clusters, and it's particularly prevalent in cancer cells. By understanding the processes involved with these diseases and understanding at molecular level how molecules interact with light, we can use that basic information to try and understand a much more complicated system, ideally in these areas of disease. I'm the first person in my family to ever even have a degree, let alone a PhD, or let alone be an academic. My dad's a plumber. In fact, when I was an undergrad, I spent most of my summers putting in kitchens as a way to pay through my undergraduate degree. And actually, those skills you learn, you think they wouldn't be linked, but actually they really are. That the ability to take your car apart, or to take your computer apart, or to take your plumbing system at home apart. We use that ability, that kind of curiosity of taking things apart and wondering how they work, is exactly what we do today, except for we're just doing it with lasers and fluorophores and single molecules rather than copper and carburetors and things. We're a collaborative lab, and we try to do public outreach events as well as social events. I'll give you an example. A few years ago, we did an installation at the Science Museum Leitz, where we did an exhibition called "How Can a Gin and Tonic Save Your Life?" If you've ever seen a gin and tonic under black light, you see it glows blue. And the reason it glows blue is because there's a molecule in tonic water called quinine, and quinine was actually the first malarial drug you may have heard of, but actually the reason why it exhibits a different property called fluorescence, and so it's absorbing the ultraviolet light and emitting blue light. And we use that same phenomenon in the lab, but instead of quinine we use different molecules, and instead of a black light we use a laser. And we're using that to try and understand the molecular origin of adaptive immunity. And so what we did is we went to the Science Museum. We had a bunch of gin and tonics. We gave gin and tonics away to people, and we got them to try and understand about super resolution and fluorescence imaging. And actually that was fantastic. It wasn't just fantastic for us to kind of communicate our science to the kind of general public, but actually we ended up engaging with a company that came to us and said, you know, this is fantastic science and you're terrible at presenting it. And we said, we agree with you 100%. And they helped us. We've been developing a software platform. We've just had a paper published where we created the first ever virtual reality software to be able to visualize some of our 3D microscopy data. And so this was fantastic, not just because, you know, it's a scientific output and we're interested in scientific output, but this only came about through doing engagement. And so this is a good example of how it's just, you know, it's not just giving back, it's a total two-way thing. And we certainly wouldn't have collaborated with those researchers and that team had we not bothered to try and give away gin and tonics. So I suppose we should continue to give away gin and tonics in the lab. That might help.